Welcome to the deep dive. Our challenge today is, well, analyzing the almost invisible. We're talking about those microscopic traces of biological stuff left behind. Maybe a few skin cells, a single hair. If you can barely see it, how on earth do you analyze it? Well, the answer, the technology that truly changed everything in forensic biology, is the polymerase chain reaction, PCR. So today, we're cutting straight to the chase. This deep dive is all based on Chapter 7 of Forensic Biology, second edition by Richard Lai. Think of it as your fast track, a really comprehensive guide to the essentials of how DNA gets copied for forensic work.
Right. PCR is basically like a molecular photocopier, but maybe more precise than that sounds. It lets you exponentially amplify a very specific piece of DNA. You might start with a tiny, tiny amount, sometimes less than an nanogram.
Yeah.
And the process gives you millions, even billions of copies. We call those copies amplicons. And those are what you can actually work with for tests like SDR profiling or figuring out how much DNA you started with.
That sensitivity, that's the real breakthrough, isn't it? It means that tiny swab from a steering wheel or a faint stain suddenly becomes usable evidence.
Exactly. Its incredible sensitivity is why it's fundamental now. The basic idea was floated back in the 70s, but it was really Kerry Mullis and his team in the mid-80s who nailed the technique initially for sickle cell anemia diagnosis. But the huge leap for automation came with using a thermostable DNA polymerase, one from a bacterium, Thermus aquaticus, that lives in hot springs.
So it doesn't break down when you heat it up repeatedly.
Precisely. That enzyme tolerates the high temperatures needed for the cycles. That's what made automated PCR possible and, well, led to the Nobel Prize in '93.
Okay, so let's break down the fundamental molecular stuff driving this. Before copying, you have to pull the DNA strands apart, right? That double helix structure. It's held together by those hydrogen bonds between the base pairs. A with T, C with G weak electrical attractions.
Yep, and also some base stacking interactions. And that process of separating the strands, that's called denaturation. Usually, we just do it by heating the solution. Nice and simple.
Okay.
And there's a key term here, melting temperature, or $CHM. That's the specific temperature where exactly half, 50%, of the DNA strands in your sample have separated or denatured.
And why is knowing that exact $10 so important for the forensic scientist running the test?
Because it's critical for setting up the PCR machine, the thermal cycler.
Yeah.
If your annealing temperature, which we'll get to, is too close to or above the $10, your primers won't stick. If it's way too low, other things might go wrong. And what affects that $10 is really interesting. A big factor is the nucleotide content. Remember, C pairs with G using 3 hydrogen bonds.
Right, whereas A and T only use 2.
Exactly. So DNA regions with a lot of G's and C's, what we call high GC content, they have more of those triple bonds holding them together. It takes more energy, meaning higher temperature, a higher $10 to pull them apart.
Makes sense, like a stronger zipper.
Kind of, yeah. Length also matters, longer dowel. DNA needs a higher $2. And the chemical environment, like salt concentration or pH, that affects it too. But GC content is a really core factor.
So after you've pulled the strands apart with heat denaturation, they need to come back together at least partially for the primers to bind. That's renaturation or reannealing.
Both terms work, yeah. Renaturation or reannealing. It's essential for the primers to find their target spots. And for this to work properly, you need a couple of things. First, you need enough charged molecules, like salts, in the buffer. They help neutralize the negative charges on the DNA's phosphate backbone. Without that, the strands would just repel each other.
Okay, reduce the repulsion.
Right. And second, the temperature needs to be just right. Cool enough so the primers can bind strongly to their complementary sequences on the template DNA. But, critically, it has to be warm enough to prevent them from binding randomly or weakly to places they shouldn't. That causes non-specific products, which is bad news. Now, this basic cycling process paved the way, in the early 90s, for real-time PCR, often called qPCR.
So instead of just seeing the result at the very end.
Exactly. Before qPCR, you ran the cycles and you check the result. With qPCR, you can actually monitor the amount of amplicon accumulating in real time during each cycle. It's happening simultaneously with the amplification itself.
And that gives you that classic S-shape amplification curve you see in textbooks. If you imagine plotting the amount of product versus the cycle number, what are the important phases on that curve?
Okay, picture that S-shape. The first really crucial part, especially for forensics, is the exponential phase. This is where the amount of product is, ideally, doubling every single cycle. And the critical point here is that when your signal crosses a certain threshold in this phase is directly related to how much DNA you started with.
So a stronger starting sample hits the threshold in fewer cycles?
Precisely. That lets us quantify the initial amount of DNA, which is vital for interpreting results later on.
Got it.
Then, as the reaction progresses, things start to slow down. You enter the linear phase. Here, the efficiency drops because one or more components, maybe the primers or the building blocks, are getting used up. And finally, you hit the plateau phase. No significant new product is being made. The reaction has essentially run out of steam. Reagents are depleted. Maybe the enzyme is losing activity.
You mentioned the relationship between starting amount and cycles needed. What's that called?
We talk about amplification efficiency. Basically, it reflects how close to perfect doubling you get each cycle in that exponential phase. A higher starting amount needs fewer cycles to reach detection. You can plot the log of the starting amount against the cycle number, and the slope tells you the efficiency.
Okay, let's dig into the toolkit, the ingredients. What's the absolute workhorse component that makes cycling at high temperatures possible?
That's got to be the thermostable DNA polymerase. The original was Taq polymerase, but for forensic work, a modified version called AmpliTaq Gold DNA polymerase is very common.
What's special about the gold version?
It enables something called hot start PCR. See, when you mix all the ingredients together at room temperature before starting the cycling, there's a risk. The primers might bind loosely to the wrong places on the DNA, or even worse, they might stick to each other.
Forming primer dimers.
Exactly. Primer dimers are useless short fragments made just from primers binding together. They compete with your actual target DNA for reagents and enzyme time. It wastes resources.
So how does hot start stop that?
The AmpliTaq Gold enzyme is chemically modified so it's inactive at lower temperatures. It's essentially switched off. It only gets activated during the very first high-temperature step of the PCR cycle, usually around 95 degrees Celsius. This heat does two things. It activates the enzyme, and it denatures the template DNA, ensuring the primers only bind when conditions are optimal.
Clever, so you avoid all that messy low-temperature activity.
Right. It greatly increases specificity and yield, which is crucial when you're dealing with potentially complex or low-level forensic samples.
Okay, polymerase is the engine. What about the instructions, the PCR primers?
Primers are critical. They're short, single-stranded pieces of DNA, typically oligonucleotides about 15 to 25 bases long. You always need a pair, a forward primer and a reverse primer. They're designed to bind to specific sequences on opposite strands of the DNA, effectively flanking or bracketing the target region you want to copy.
And designing them correctly sounds like a bit of an art.
It's definitely a science. There are strict rules. First, they must be specific only to your target sequence, no binding elsewhere. Second, the forward and reverse primers in a pair need to have very similar melting temperatures, their Tonneval values. Usually you want them within 5 degrees Celsius of each other.
Why is that similarity so important?
Because both primers need to bind effectively during the same annealing step in the cycle. If one primer's ideal binding temperature is much different from the others, you'll compromise the efficiency for one or both, and the reaction won't work well.
Makes sense.
Other rules. GC content should ideally be around 40 to 60%. And, crucially, you have to design them to avoid sequences that are complementary to themselves.
Ah, so they don't fold back and stick to themselves.
Right, that forms what we call hairpin structures.
Yeah.
Also, you need to avoid sequences that are complementary to the other primer in the pair, beyond their intended binding sites, to prevent those primer dimers we talked about.
So a lot of checks involved in good primer design.
Absolutely essential. Bad primers mean bad PCR or no PCR.
Now, in forensics, we're usually not looking at just one DNA region. STR analysis looks at many locations, maybe 20 or more. You can't run 20 separate PCR tubes for every sample, can you?
No, definitely not. That's where multiplex PCR comes in. It's a technique where you put multiple primer sets for all those different STR regions, for example, into the same reaction tube. So you amplify many different target regions simultaneously in one go. It's vital for the high-throughput workflow in forensic labs.
What's the catch? It sounds complex to get all those primer pairs working together.
The main challenge is compatibility. All the different primer pairs in your multiplex reaction need to have similar annealing temperatures and similar amplification efficiencies. You don't want one SPR locus amplifying really well while another one barely works because the primers aren't optimized to work under the exact same conditions.
Yeah.
It requires very careful design and optimization.
Okay, so we have the polymerase engine, the primer blueprints. What other raw materials and conditions are needed?
Well, you need the template DNA itself, obviously. For standard forensic tests, labs often aim for about 1 to 2.5 nanograms. You need the building blocks, the 4-deoxynucleoside triphosphates, or dNTPs. That's DATP, DCTTP, DGTP, and DTTP. The polymerase uses these to build the new DNA strands.
The A's, C's, G's, and T's.
Exactly. And you need the right chemical environment. That means a buffer solution to keep the pH stable, usually between 8.3 and 8.8. And critically, you need divalent cations, almost always magnesium ions, Mg2++, MBIS. The polymerase enzyme absolutely requires magnesium to function correctly. Sometimes monovalent cations like potassium, a color plus, are included too.
So a carefully balanced chemical soup.
Pretty much. Every component matters.
And to make sure that soup is cooking correctly, you run controls.
Always. Controls are non-negotiable. A positive control uses a known standard DNA sample. If the positive control works, it tells you your reagents are good, your thermal cycler program is correct, everything is basically functional.
And the negative controls.
We use those mainly to check for contamination, which is a huge issue we need to discuss. There are typically extraction region blanks and amplification negative controls.
We'll definitely circle back to contamination. But first, let's put all these ingredients into the machine and run the actual thermal cycle. Describe those 3 key temperature steps.
Okay, it's a repeating cycle. Step 1 is denaturation. You crank the heat up high, typically 94 to 95 degrees Celsius. This separates the double-stranded DNA template into single strands.
Step 2.
Annealing. You drop the temperature significantly. The exact temperature depends on the primer's $, but it's usually set about 3 to 5 degrees Celsius below the calculated $, of the primers. This lower temperature allows the forward and reverse primers to find and bind or anneal to their specific target sequences on the separated DNA strands.
And you mentioned this annealing temperature is critical. What if it's slightly off?
Yeah, it's a fine balance. If the annealing temperature is too high, the primers won't bind efficiently. Maybe they'll fall off too easily. That leads to low product yield.
Okay.
But if it's too low, the primers might get sticky and bind non-specifically to sequences that aren't their perfect match. That generates unwanted PCR products and messes up your results.
Right, precision is key. What's the 3rd step?
Extension, sometimes called DNA synthesis. You raise the temperature again, usually to about 72 degrees Celsius. This is the optimal working temperature for the Taq polymerase enzyme. The polymerase finds the bound primers, latches on, and starts adding dNTPs, synthesizing a new DNA strand complementary to the template strand. It extends from the primer.
Denature, anneal, extend, and then repeat.
Exactly. That 3-step process is one cycle, and you repeat it typically 25 to 35 times.
And with each cycle, the number of copies of your target sequence nearly doubles. That exponential amplification is just mind-boggling when you think about it.
It really is. The theoretical number of copies after ACT cycles, starting with $n copies, is roughly 6-andle 1 plus ESE, where E is the efficiency, ideally close to 1.
So run just 28 cycles, assuming good efficiency, and you can get something like 100 million-fold amplification.
Yep. And push it to 34 cycles, and you're potentially looking at a 10 billion-fold an increase, man.
Wow.
That explains why PCR is so incredibly sensitive, but also why even tiny issues get magnified. And it highlights why the number of cycles chosen is really important, especially for samples with very little starting DNA.
Which brings us neatly to the major challenges faced in forensic PCR. What are the big hurdles?
Well, the first big one is template quality. DNA from crime scenes isn't always pristine. It's often degraded, meaning the DNA strands are broken into smaller fragments due to environmental exposure, like sunlight, heat, or bacteria.
And if the break happens right in the middle of the STR region you're trying to copy?
Then the PCR just fails for that target. The polymerase can't bridge the gap. This is why longer target regions, longer amplicons, are inherently more likely to fail in degraded samples compared to shorter ones.
Makes sense. What's another major quality issue?
Low copy number, or LCN. This refers to samples where you have very, very little starting DNA. Maybe just 100 picograms or even less, which is only about 15, 20 cells worth of DNA.
And what problems arise when you have so little DNA?
The main issue is the stochastic effect. Stochastic just means random chance is playing a big role. Imagine a person is heterozygous at an STR locus, meaning they have 2 different alleles, say allele 10 and allele 12. If you only manage to get, say, 5 copies of the DNA containing that locus into your PCR tube just by random chance, you might happen to grab 4 copies of allele 10 and only 1 copy of allele 12.
Okay.
When you run the PCR, allele 10 gets amplified strongly, but the single copy of allele 12 might amplify poorly or not at all. It fails to reach the detection threshold.
So the result looks like the person only has allele 10, a false homozygote.
Exactly, you get allele dropout. Or sometimes you might see a huge imbalance between the 2 allele peaks. These stochastic effects make interpreting LCN profiles really challenging and increase the risk of errors.
That seems like a huge potential pitfall. How do labs try to deal with LCN issues?
One common strategy is simply to increase the number of PCR cycles. Instead of the standard 28 cycles, maybe they run 32 or 34.
Trying to give those low-level alleles more chance to get amplified up to detectable levels?
That's the idea. More cycles can help overcome dropout, but it also increases the risk of amplifying any tiny bit of contamination that might be present, so it's a trade-off.
Okay, so degradation and LCN are quality issues. What about things in the sample that interfere? Inhibitors.
Ah, yes, inhibitors are a constant battle. These are substances present in the original biological sample or introduced during collection that interfere with the PCR process itself. They might bind to the DNA template, making it unavailable, or they might directly inhibit the DNA polymerase enzyme. Either way, the PCR fails or performs poorly.
What are some common culprits found in forensic evidence?
Oh, there are many. Heme from hemoglobin in blood is a classic inhibitor. Indigo dyes, like from denim jeans, are notoriously problematic. Melanin pigment from hair samples can inhibit PCR. Soil, textiles, cleaning chemicals, the list goes on.
How do labs fight back against inhibitors?
The first line of defense is during DNA extraction, trying to purify the DNA away from these substances. But sometimes inhibitors carry over. If you suspect inhibition, you can try a few things. Sometimes simply diluting the DNA extract helps, although that's counterproductive if you already have an LCN sample. You can use special cleanup columns or centrifugal filtration devices that physically remove inhibitors based on size.
Or can you tweak the PCR chemistry itself?
Yes. Sometimes increasing the concentration of the DNA polymerase in the reaction can help overcome the inhibition, kind of like adding more soldiers to the fight. Another very common strategy is to add bovine serum albumin, or BSA, to the PCR mix. BSA is thought to act like a sponge, binding up many common inhibitors and preventing them from interfering with the polymerase.
Interesting. So quality issues, inhibitors. What's the third major headache?
Contamination. Because PCR is so incredibly sensitive, amplifying tiny amounts of DNA billions of times, even the smallest amount of contaminating DNA can be amplified and potentially swamp the actual evidence sample.
Where can contamination come from?
Everywhere, potentially. From the crime scene itself during evidence collection, from lab personnel during handling, from reagents, pipettes, tubes, even from previous PCR products aerosolized in the lab air. Carryover contamination.
So prevention must be absolutely paramount.
Absolutely. Strict protocols are essential. Labs have dedicated pre-PCR and post-PCR areas, often physically separated with equipment, pipettes, reagents. Lab staff wear protective gear, lab coats, gloves, masks, hairnets. They use special aerosol-resistant pipette tips. All solutions and tubes should be certified DNA-free. It's meticulous work.
And those negative controls we mentioned earlier, they help track it down.
Yes, they're crucial for monitoring. The extraction region blank goes through the whole process from extraction onwards, but with no sample added initially. If DNA shows up in this blank, it means contamination likely happened during extraction or possibly reagent prep.
And the other one?
The amplification negative control. This one contains all the PCR regions mixed together, master mix, primers, water. But no template DNA is intentionally added. It's set up alongside the actual samples. If this control shows a DNA profile but the extraction blank is clean, it points towards contamination occurring specifically during the PCR setup step itself.
And labs usually have DNA profiles of their staff on file.
Yes, that's standard practice. If contamination is detected in a controller sample, comparing it to the staff database can often identify the source, helping to pinpoint and fix the contamination issue.
Okay, that covers the main PCR challenges. Let's shift gears slightly. We usually think DNA and RNA badge protein, the central dogma, but can information flow the other way?
It certainly can. The process of using an RNA molecule as a template to synthesize a complementary DNA strand, or cDNA, is called reverse transcription. It was discovered back in 1970 by Baltimore and Timmon working with retroviruses. The enzyme that does this is called reverse transcriptase.
And if you combine reverse transcription with PCR, you get RT-PCR. Why would forensic scientists want to look at RNA when DNA is usually the target?
The primary forensic application of RT-PCR is for bodily fluid identification. Different tissues and cell types in the body express different sets of messenger RNA, or mRNA. So, for example, blood cells have specific mRNAs that aren't found in saliva. Saliva has its own unique mRNA profile, different from semen, and so on. By using RT-PCR to detect these tissue-specific mRNAs in a crime scene stain, you can determine what type of bodily fluid it is, which can be really important context.
Ah, so it adds another layer of information beyond just who the DNA belongs to. How does the reverse transcription part actually work? Do you need primers for that too?
Yes, reverse transcriptase needs a primer to get started, just like DNA polymerase. Interestingly, DNA primers tend to work more efficiently than RNA primers for initiating reverse transcription. The enzyme then reads the RNA template strand and incorporates the dNTP building blocks to synthesize the complementary DNA strand, the cDNA. Many reverse transcriptase enzymes also have an intrinsic RNase H activity, which degrades the original RNA template strand after the cDNA has been made.
Are there specific types of reverse transcriptase enzymes used?
Yeah, labs often use genetically engineered versions, commonly derived from viruses like avian myeloblastosis virus, AMV, or Maloney-Marine leukemia virus, MLV. Some modern ones are designed to be stable at higher temperatures, maybe up to 60 degrees Celsius.
Why the higher temperature stability?
RNA molecules can fold up into complex secondary structures, like loops and hairpins, due to internal base pairing. These structures can block the reverse transcriptase. Running the reaction at a higher temperature helps to melt these structures, keeping the RNA template linear and accessible, which improves the cDNA yield.
What kinds of primers do you use for the reverse transcription step? Are they specific to the mRNA you're looking for?
You have options. You can use gene-specific primers, GSPs, that target only the particular mRNA you're interested in. But often, especially if you want to analyze multiple markers later, labs use universal primers. One common type is oligo-DT primers. These are short chains of thymine bases that bind to the polyadenine, the poly-A tails, found at the end of almost all eukaryotic messenger RNAs. So they prime cDNA synthesis from most mRNA present.
Okay.
Another option is random hexamer primers. These are short primers, just 6 bases long, made up of random sequences. They can bind pretty much anywhere along any RNA molecule in the sample, including mRNA, but also ribosomal RNA. This gives you cDNA representing a broader range of the RNA population.
So once you've made the cDNA, you then do the PCR part. Can this all happen in one tube?
It can. That's called one-step RT-PCR. You put the RNA template, reverse transcriptase, DNA polymerase, primers, which must be gene-specific in this case, dNTPs, and buffers all together in a single tube. The thermal cycler runs a reverse transcription step first, then transitions directly into the PCR cycles. It's simpler, fewer pipetting steps, less chance of contamination between steps.
What's the alternative?
Two-step RT-PCR. Here, you perform the reverse transcription reaction in one tube first. This is where you can use those universal primers like oligo-DT or random hexamers to convert lots of different mRNAs to cDNA. Then you take a small portion of that resulting cDNA mixture and add it to a separate tube for the standard PCR amplification, using gene-specific primers for the targets you want to detect.
Which approach is more common in forensics?
Forensic applications, especially for body fluid ID, looking at multiple mRNA markers, typically use the two-step RT-PCR approach. It offers more flexibility because you create a cDNA pool first using universal primers, and then you can use aliquots of that pool to run separate PCRs for different tissue-specific markers. And often, the final detection isn't real-time qPCR, but rather endpoint PCR. You run the cycles, then analyze the final PCR product, often using fluorescently labeled primers and capillary electrophoresis, similar to STR analysis.
Wow, okay. We have definitely covered a huge amount of ground there. Just to recap the main points, PCR is this incredibly powerful, rapid method for making billions of copies of DNA, all based on the fundamental processes of denaturation and annealing. We talked about the key ingredients, the thermostable polymerase like TAC, the critical role of primer design, and the need for controls.
Right, and we really dug into the major hurdles in forensic work, dealing with degraded or low copy number DNA and the associated stochastic effects, battling inhibitors from the evidence itself, and the constant overriding need to prevent and monitor for contamination.
Success really hinges on incredible precision in temperature control, in primer design, in lab practice.
Absolutely. And we saw how RT-PCR cleverly extends this core technology. It lets us move beyond just DNA identity to analyze RNA, giving valuable clues about the type of biological material present, like identifying blood versus saliva. It connects that core molecular biology right back to interpreting the crime scene evidence.
So, considering PCR's almost unbelievable power, making 10 billion copies from potentially just a handful of starting molecules, here's a final thought for you to ponder. How might forensic tech evolve further? Could we move beyond just detecting DNA's presence, maybe towards measuring subtle variations, perhaps in polymerase efficiency itself when faced with trace inhibitors, or slight differences in amplification kinetics to build even more detailed, context-rich profiles from evidence in the future? Something to think about.